another 5 minutes because the schedule time is 10. So
Okay, sir.
I guess
Okay, no problem.
it will be 9. After 5 minutes, people join. Then it will be better for us. Hello, Kushal.
Yes, sir.
Hello. Kushal,
sir.
এখানে participant list বা এটা কিছু দেখাচ্ছে না। As
Sir,
such.
attendance list আমি দেখতে পাচ্ছি 74 attendees still now joining in this.
আচ্ছা না, actually I was asking for whether director sir or any other they have already joined or not. আমার তো সেটা
স্যার মিউট হয়ে গেছে।
হ্যাঁ
এবার ঠিক আছে?
হ্যাঁ হ্যাঁ।
ওকে, তো জানতে পারলে ভালো হতো।
স্যার সন্দীপ ওরা চেক করতে থাকছে যে স্যার যদি জয়েন করে ওরা প্যানেলিস্টে তুলে নেবে।
ওকে, না এখানে অ্যাকচুয়ালি কোনো লিস্ট শো করছে না আমার স্ক্রিনে, আমি মোবাইল থেকে করছি।
হ্যাঁ, যারা কোহোস্ট তারাই দেখতে পাচ্ছে। তো ধ্রুবজ্যোতি স্যার হোস্ট আছে, ঠিক আছে, ওনাকে আমরা বলছি সবাইকে কোহোস্ট করে দিতে।
সেটা বেটার হবে। ওকে ওকে, ঠিক আছে।
আমি দেখতে পাচ্ছি।
ওকে, একটু তাড়াতাড়ি করতে বলবি, হ্যাঁ, তাহলে আমি ব্যাপারটা পুরোটা ফলো আপ করতে পারবো।
Sir, should I start now? Should we start now, sir? Hello.
কোহোস্ট করেছে কি?
হ্যাঁ স্যার, আমি হোস্ট করা হয়েছে।
Yes sir, everyone is co-host.
Okay.
Dhruv sir, should we start now?
Good morning to all of you.
Okay, sir.
Good morning, sir.
Techno India University is happy to invite all of you in the second webinar. So today is the second day of the webinar. Let me introduce Supradeep Mondal. In short, he is famous as SM in our department. SM sir. He is the head of the Department of Pharmacognosy and Phytochemistry department, and my very good friend, philosopher, guide, everything. So, Supradeep, you please start the introduction and welcome speech.
Yes, sir. Good morning, one and all. Myself, Supradeep Mondal, faculty of School of Pharmacy, Techno India University, West Bengal. So it's my privilege to welcome Dr. Rutesh Vyas in today's session. So we are in the second day or final day of our today's webinar program. So Sir, Dr. Rutesh Vyas is the postdoctoral research fellow of Duke University, Connecticut, USA. So he started his career from North Gujarat University. He completed his B.Pharm in the year 2005 from Sri Sarvajanic Pharmacy College under North Gujarat University. Then he has done Masters in Pharmacology and Toxicology in the year 2009 from AMS College of Pharmacy, Long Island University, New York, USA. Then he has done PhD in molecular biology and biochemistry in molecular biology and biochemistry from Wesleyan University, Connecticut, USA in the year 2017. So he is having a huge research experience. He has worked in the field of diabetes and diabetic nephropathy in CVS and metabolic disorders. He has also worked as a research assistant in the Department of Hematology and Oncology in Children's Hospital, Boston. He has worked as associate in research in the Department of Cell Biology, Duke University, School of Medicine, North Carolina. Then he has worked as a postdoctoral research associate in the Department of Cell Biology, Duke University, School of Medicine, North Carolina, and then University of Postdoctoral Fellow in the Department of Neuroscience, Yukon Health Center, Connecticut. So, sir, it's my privilege to welcome in the session as the first speaker of today. Good morning, you. Good morning to all of us. So, sir, should we start the session with the due permission?
Yes, definitely.
Okay. Thank you very much.
Good morning, Dr. Rutesh Vyas.
Yeah, thanks very much, Dr. Mandal. And thank you so much to Dr. Sen.
Sir,
as
Mr.
well.
Mandal, sir, sorry for interrupting Mr. Mandal, Supradeep Mandal.
Yeah, Dr. Mandal, right?
Mr. Mandal, not
Oh,
doctor.
Mr. Mandal. Okay, all
Going
right.
to be doctorate very soon.
Okay, all right,
So
nice.
it is advanced booking. This is advanced booking.
Advanced booking,
Okay.
yeah, advanced
Yes.
compliment.
Okay, thank you so much.
Right, it's been my very much pleasure to to be here on this platform with all of you guys, and I can already see 99 participants, so it's a huge crowd. Very excited about it. And today I'm here to share basically my journey from North Gujarat University to where I am right now as a postdoctoral fellow, and we will be skipping. I won't be able to talk about the Pfizer work because of conflict of interest and some other work. But I will cover three major stories, and I hope you will like it. I was a very ordinary student, in Mehsana, very small village, small town in North Gujarat University, and I basically started my career under the guidance of Dr. Sen, where I basically, do you guys see my screen? Sorry to interrupt, but let me see. You guys
Yes,
see
sir,
my
it is clearly visible.
Okay. All right. Fantastic. So basically, the seeding process to instigate my research interest and curriculum was seeded by Dr. Sen, and he's a very parently, kind of figure to me so far. And after so many years of living Mehsana and coming all the way here, it's been very fantastic support in my career development. So I'm very, very, very thankful to have Dr. Sen as my mentor, my ex-mentor, and yet my mentor as well for the rest of my life. So we'll be covering three major stories. So currently, I'm in a transition process from the Duke University Health Center to UConn. It's one of the state agency in the upstate, in the New England area of the USA, Farmington. And I'll be talking to you mostly about my Boston Children's Hospital work, my PhD work, and my postdoctoral research work, and some of the collaborative work. So my major area of interest was olfaction, neurogenesis, and how we might implicate these modalities into pathogenesis, which is a stroke related. So this is the highlight of my talk. I will be covering, as I said, 3 stories. The part 1, in which I will be talking about overcoming reprogramming resistance to Fanconi anemia, the work done
Dr.
at
Vyas,
Boston Children's Hospital.
Dr. Vyas, just 1 minute.
Yes.
Sorry to interrupt you.
Yep.
Let me introduce first.
Yes.
Let me introduce first. Okay.
Sure.
Then you
Yeah.
start your talk.
Go ahead.
Okay. So today's first speaker is Dr. Rutesh Vyas. Rut means season, which is changing, and Ish means Ishwar, means Lord. So the total meaning of Rutesh is Lord of Truth and season, which is changing from time to time. Now I am starting from his past days at SSPC, Sri Sarvajanik Pharmacy College, Mehsana, the year 2005, the long, long year from this, around 15 years ago. He was in the final year, B Farm student. He became Mr. Vyas to Dr. Vyas and then postdoctoral research scientist at USA. In 2005, he came to me when he was studying at final year B Farm student in Sarvajanik Pharmacy College, North Gujarat University, Mehsana. And he came to me and told me, sir, मुझे research करना है, एकदम fantastic, mind-blowing research. Then he started his research journey of research, and he collected so many industrial effluents from industry, and he did a fantastic job. And his project was industrial effluent follows the law of conservation of energy, and he has presented this paper, research paper, in annual convention of chemists and selected for Young Scientist Award in the year 2004. Then he did his pharmacological research on blood hematological aspects. His paper, Pharmacological Exposure Towards Hematological Factors, enhances a new approach on bioavailability, and he presented this paper on national conference in Lucknow in 2005. Then he started his research work on Caesalpinia crista seeds. I think you remember this. This is long, long background, nostalgic effort. Antimicrobial screening of Caesalpinia crista seeds, and he did a fantastic research work which has been published in Journal of Advances in Biological Sciences in the year 2005. Then he did another project on biological screening of pydazolo pyrimidine nucleus adduct with indane moiety for COX-1 and COX-2 inhibition, and his research work has been accepted in International Conference on Materials Chemistry MC7 in United Kingdom, Edinburgh University in 2005, and he went there to present this paper. Before going to UK, he visited my house. He was coming from Ambaji with his parents and said that I have taken 2 blessings, one from goddess Ambaji and another from me before leaving to UK, University of Edinburgh, and he has presented his research work very fantastic way in that I got that report from United Kingdom, and this uh, conference has been sponsored by Royal Society of Chemistry, England. His research work on Caesalpinia crista seeds was very much fantastic and showed highly potent antimicrobial activities. In Sarvajanik Pharmacy College, he did this research project. After completion of this B.Pharm course, he got chance in Master in Pharmacology and Toxicology in USA, and his advisor was Dr. Mrinal Bhattacharya. And thesis work was minimum sequence homology requirement for allelic exchange in Actinobacillus actinomycetimicans in understand natural transformation and mutagenesis, and he did his master's in 2006 to 2009. Then he got chance in PhD in molecular biology and biochemistry and finished his work in between 2011 to 2017 under Dr. Robert Lane at University of Wesleyan, Middletown, Connecticut, and his dissertation topic was molecular and epigenetic regulation of olfactory receptor gene choice. After completion of his B.Pharm course, he did his industrial training at Ahmedabad Intas Pharmaceuticals at Biotechnology and Research Center, Ahmedabad, 2004, May to 2000 August 2004. Then he did his one work on hematological aspects, and he has published his paper on this. Then he worked as research associate in the Department of Pharmacological and Toxicological Institute and Zydus Research Center, 2005 to 2006. Then he went USA and he did his masters. Afterwards, he did his doctorate, and then now he is doing postdoctorate research under the supervision of Nobel laureate. And I have given one recommendation for this, and he got chance under Nobel laureate postdoctoral research scientist. His biodata is very big, and it is at least 7 to 8 pages. In short, it is Dr. Rutesh Vyas is a postdoctoral fellow in Pharma Lab, USA. He is investigating a molecular, biochemical, epigenetic, proteomics, and behavioral aspects of the acute and chronic stroke. He is also interested to study developmental and injury models of neurogenesis versus astrogenesis. Towards this goal, he will be investigating novel mechanism of neuroprotection in the CNS. His research area is very vast. For that, he will be looking at factors from acute and chronic peripheral vasculature as well as local and neurogenetic niche specific injury response. Dr. Rutesh has completed his PhD from Wesleyan University, and his first postdoctoral training was from Duke University. He has worked at Pfizer Global Research Laboratories on cardiovascular and metabolic disease and Boston Children's Hospital in hematology and oncology at USA. So this is the big journey of Vyas, who became Mr. Vyas to Dr. Vyas, and then Dr. Vyas to postdoctoral research scientist at USA. And I am very proud that he is my very favorite student. And his today's topic is isolation, characterization, and activation of patient neuronal stem cells in the SVZ niche under topic neurogenesis and stroke. So this is his vast research area, which is compiled with so many research aspects, and I am happy that all of you will listen his research topic and will be very happy. So Dr. Vyas, you can start your research projects. I am very happy that you became Mr. Vyas, B.Pharm, then M.Pharm, Masters, then doctoral studies, Dr. Vyas. Then now he has taken his duties as postdoctoral research fellow at Duke University, and I am very proud that Dr. Vyas is now with us with his nice, excellent talk. So, Dr. Vyas, you can start.
Okay, sounds good. Thank you very much, Dr. Sen, for a very kind and very nice introductions. And you've always been my excellent mentor and parently figure. So thanks for supporting throughout this journey. So raising the hand, meaning, do I need to answer any question right now, or you can post your question and answer in the chat box if you like. Or just stop me if I'm going too fast, or if you guys have any questions. So, but I'm going to jump directly into my second slide, which is the highlight of my talk. So I'll be covering three major stories of my research areas. The first one was performed at Boston Children's Hospital. The second one is my PhD work, and the third one is basically my collaborative postdoctoral research work, where we, this is the one of the mature story that I could talk about right now. There are several other under development stories which I won't talk about. So one of the first thing that I learned while as a research assistant in Boston Children's Hospital is gene therapy and preclinical gene trials to develop the gene therapy. And which is quite interesting to me because, you know, as we can see here, that there is this Fanconi anemia was a model system that we were trying to develop the gene therapy for. And I was assisting 3 independent postdoctoral fellows right after my master's when I became a master, graduated from the master degree from New York. This was my second job right after the Pfizer pharmaceutical. And so what happens in Fanconi anemia is basically there is a growth retardations. As you can see that this patient right here, and this is the congenital X-linked recessive disorder, and mostly it's associated with the birth, the birth or the post-birth defect, where there is a short stature. The fingers, as you can see, the toes are very abnormal. And if you look deep into the bone marrows, that bone marrows are not very well developed, and there are bone marrow stem cell failure in the bone marrows, and they acquire 1,000-fold higher risk of cancer development. And as you can see that if you look into the genetic pattern, there is a cross-linking of their chromosomes. So there is a chromosomal abnormalities which can break and predispose to the cancer conditions, and their blood production is somewhat abnormal as well. They could not sometimes make the proper proportions of all the different blood lineages, myeloid and the other erythroid lineages. So this is a major difficulty in the US as well as all across the world. This is a genetic disease. And so if we look deep into the molecular pathways, so here is a Fanconi anemia DNA repair pathway and cancer. So whenever there is a DNA damage or break, there are ATR kinases, that gets activated, and they phosphorylate this so-called complex 1. Complex 1 is nothing but a 8 different gene of Fanconi anemia clustered together. And it activates basically the FANC-CD2, which is another Fanconi anemia key gene. The ubiquitinations of that FANC-D2 would then activate the complex 2, which is a BRCA repair pathway, okay, which is one of the major DNA repair pathway. And once the damaged DNA is repaired, there is a deubiquitinating enzyme called USP1, which will deubiquitinate the FANC-D2 loci, deubiquitinations, and then FANC-D2 will then recycle. So this is how the DNA repair would function normally. The mutations in any of these Fanconi anemia gene, FANC-A, G, B, C, F, E, and L, or D2, would lead to genetic abnormalities and will lead to Fanconi anemia. So complementations of those genes would then become necessary, and it's a provide you the hot target for developing the clinical gene therapy. So these are the biomarker for both the functional as well as the protein and gene expression level. So if you look at the immunofluorescence for the BRCA1 and FANC-D2, where the Fanconi anemia pathway is intact, so we are looking at a single cell nucleus, and this green spot is a FANC-CD2 foci, stain in green, labeled with the green antibodies. And if you look into the Fanconi anemia, where the pathway is defective, they do not develop any FANG CD2 repair foci as clearly indicated in this right panel, right cells. Also, if you look at the Western blot, there is a single band in both Fanconi anemia pathway intact population of cell as well as Fanc anemia pathway defective cells. But if you look here into an intact pathway, there is a higher molecular weight band, which is due to the Fanc gets ubiquitinated, and whereas that particular band, high molecular weight band, is absent, where the pathway becomes defective. So this suggests that the pathway is really functional. And if you look at the protein and gene expressions, right, then this is the immunohistochemistry image where the dark brown color nuclei in several cells shown here are the Fanconi anemia protein. And then the protein also gets lost in the Fanconi anemia patients. Also, if you look at the mRNA level, so there is a shift towards the right-hand side, where there is a 8-fold reductions into the production of the FANG gene. So this suggests that they definitely, if you try to amp up or bring it back to the normal level, either at mRNA and thus the protein level, maybe you should be able to control somehow. So how one can bring it back to the normal level? So there have been efficacy studies for the molecular gene therapy in the animal model, at least. When I was doing this study, and there is a short-term and a long-term repopulation defect, as you can see here. So there are in the bone marrows, there are both short-term and a long-term CD34 positive population, so-called hematopoietic stem cells, and those populations needs to generate the blood. Various lineages of the blood cells every several time point. But because of the Fanconi anemia gene defect, there is a repopulation defect of those cell type as well. And if you look into this flow cytometry histogram, where you can see that this is a normal level required, but where the FANC-C gene, both the allele is knockout, right? So if this is FANC-C knockout, where you only have a 18% of short-term and a long-term repopulations, whereas if you try to complement the mice by putting back the correct version of FANC-C gene, so double positive, right, 2 alleles, you could rescue this defect to up to 71%. So there is a good efficacy for the therapy, gene therapy, it is proven in the mice. Now, based on this data, there have been several human clinical trials, and there are each one of these represents several clinical trials where you can see the CD34 positive cells. So what happens basically is that from the allogenic transplant, you collect the CD34 positive cells, you bring them outside, you hit them with the viruses that expresses the correct version of abnormal gene, and then you try to expand those cells and then put them back, right? But that somehow in every clinical trial, this was expected values shown in a gray, predicted values, but the actual collection of those cells were pretty low, below the threshold or required line, dashed line. So then, therefore, there was a requirement to find the alternative route at that time, that how can we make more CD34 positive cells so that, that at least the cell, for the cell therapy, the cell number is not rate limiting, okay? So in order to support that idea, there was a very famous technique at that time that was developed by George Q. Daly, which is the lab downstairs in the same building. And what they showed is basically you could take the fibroblast cells and you could reprogram them into any cell type you want. So the original research was done by Sir John Gordon and Shinya Yamanaka from Japan, and they got a Nobel Prize for generating these IPS, induced pluripotent stem cell. That means you could take any cell type. At least they took the fibroblast and they were able to push them back onto their epigenetic landscape to generate somewhat stem-like property cells. So we use the similar technique to generate more CD34 positive cells. So here, what I'm showing you is the efficiency of iPSC colony generation. So number of iPSC colony per 100,000 cells seeded. In a wild type where you don't have any defect of any gene, that means all Fanconi anemia genes are intact and they are wild type, you get a very high efficiency of induced pluripotent stem cell when you hit them with the Yamanaka factor, which is a very standard 4 virus cocktail, 4 transcription factor cocktail. And but when you try to do the same experiment with the defective initial fibroblast cells, where in this case the fibroblast is defective for FANC-C gene, both the allele are gone, and in this case it was FANC-A null. And when you hit them with the Yamanaka factors, their efficiency was somewhat reduced, I mean significantly reduced in some of the genes. Okay, and but whatever was remaining, we took those cells and we injected them subcutaneously into the recipient mice, and they were able to generate these all 3 germ layers, okay, which is a very standard in vivo test for testing the iPSC quality. So at least whatever was remaining was very good to be able to generate, and not only 3 germ layers, but when we injected into the blastocyst, we were able to successfully generate these chimeric mice with the gray and the white fur. Okay, so there was a germline contributions. Now the only matter was how can we bring these number higher. Okay, in order to do that, what we did is basically first of all we needed to find out what is causing the problem. Okay, what is causing this significant reduction in IPS generation. If we would be to go route IPS via CD34 and then the blood production. So here is a control GFP vector. At least 3 cells are shown, and here there is a OSKM 4 Yamanaka cocktail, and as you can see that when you hit them with the virus, 4-in-1 virus, there were several foci, and these foci were representing that there was when the virus integrates, they are leaving some integration DNA damage or something related to that. And when we count the percentage of cells with the FANC-CD2 foci, in the case of OSKM, there was a higher percentage of FANC-CD2 foci. That means there was a higher number of DNA damage. Okay, and when you look at the DNA damage response, it was also increased when you do have the OSKM, the Yamanaka factor, as compared to when you don't have it. Okay, in P53 increase, P21 similarly increase when you do have this Yamanaka factor, and P19 is also increased. So this is the DNA damage response was increased. And when we look at the reactive oxygen species, it was also increased. So when you have a DNA damage, you of course have all of these marker that would increase. One of the major things that we also observed is compared between the wild type and a FANC-A deficient fibroblast is that the senescence was also increased. And so these are the defect, and those needs to be well taken care of before anybody would be to go ahead and develop the gene therapy. Okay, so then what we thought is that maybe because it's happening only into the cells, those are defective in the Fanconi gene. So how about if we complement the cells with the FANC gene first? If we complement the fibroblast with the correct version of FANC gene, and then we try to reprogram, and let's see what happens. So that is exactly what we tried to do. So what we did here is we complemented. So we provided the correct version. Okay, in this case where there was a null. Okay, and we bring up the percentage senescent cells. Senescent cells was reduced actually. Okay, as compared to no complementations, and they were somewhat closer to the wild type. Not exactly, but at least they were significantly reduced. And the percentage or the number of IPS colony generation was increased. Okay, as compared to when you don't have a complementation versus when you do have a complementation of correct version. And the same quantification is shown here in a figure where you can see this is alkaline phosphatase assay, which is a very standard assay, initial assay to look for the colony in the petri dish. This individual spot represents the induced pluripotent stem cell colonies generated from the fibroblast of either the wild type mice or the mice tail tip fibroblast that is deficient in the FANG gene, okay, or the corrected version of Fanconi anemia. And you can see that there are higher colonies as compared to when you don't have a complement, okay, which further support the statistical analysis, the quantification here. And what you see here is basically increase in the fetal liver hematopoiesis. So fetal liver is one of the place where people would normally look for the hematopoiesis evidence. And so that's exactly what we did. In the fetal liver hematopoiesis, and as you can see, when you do have a complement, your fetal liver hematopoiesis was increased as compared to when you don't have, when you don't provide with the complement. So there was a better contributions also to the blood production as well. And then later on, we tried to look for the quality of these iPSCs. As you can see, these are immunofluorescence of NANOG, SSCA, TRA161 marker, OCT4, SSC4, and TRA181 with the nuclear staining shown here as a Hooks nuclear staining. Okay, and so they were very gold standard. The molecular marker for the IPS colonies. So we confirm the pluripotency, pluripotency marker. We also took these colonies, we homogenized the colonies, make the single cell suspension and injected them subcutaneously to recipient mice. And we were successfully to produce the teratoma formations. And here is a normal donor. This is a FANC anemia patient cell line where you don't see the foci, but when you do complement with the FANC gene, you start to see the foci. That means the repair pathway was rescued with the complementations. Okay. And then later on, we look for the various differentiations of the various blood lineage. In this case, colony forming unit of granulocyte, erythrocyte, colony forming unit, granulocyte, macrophage, and erythroid burst forming unit, and things like that. And their quality also looks very good. Okay, so the colony forming unit was also somewhat increased. However, it could not reach to the wild type normal donor level. But this, always in gene therapy, 5 to 10% or even 1% blood productions, the correct version of blood production is very beneficial to patients. So that's what we were expecting. Okay, so now for the first part, if you guys have any questions, let me visit the chat box. And so far I don't see any question, but you could, I can wait here before I start into my second part if you guys have any question for the first part. So that was all about the gene therapy, which is where I gather the gene therapy, the vector construction, the virus productions and things like that, all those experience, transplantations, the complementation assay, and things like that. Do I have any question yet? I don't think so. So I'm going to move on to my second part, which is the role of LSD1 and G9A in the olfactory receptor gene choice and differentiation. So this is my PhD project, and I'm so much lucky to take upon the question that was out there in the field since 2005, since the largest super gene family was identified or investigated by Dr. Richard Axel and Linda Buck, who got a Nobel Prize for identifying the largest gene family in the rat genome. So olfactory gene is one of the rich cluster of gene. Every there are at least 2,000 genes in the mice, and there are several of them in the humans. So one in every 20 gene represents olfactory receptor gene. Okay, so it's one of the largest gene family, and this is the organizing principle and the order and receptor specializations. Okay, so this is a completely different field from the field that I was working on at Boston Children's Hospital, and this was somewhat related to the neuroscience. So this was the beginning of my neuroscience training. Okay, so in this cartoon, what we have is a human, human cartoon. Human nose is smelling some flower smell, and this small colored dot represents the odorant molecule, which is a chemical molecule that is being inhaled through the nose. Okay, and so when we cut open the nose of an animal, any mice or anything, so what you see here is basically olfactory receptor epithelia. Okay, so there is olfactory epithelia. In this olfactory epithelia, there are these bipolar odorant receptor neuron. This is the only kind of neuron that would directly communicate the environment. In a single synapse, it connects to the brain, which is shown here as the olfactory bulb, which is the first relay station. Okay, through the bony skull, it passes through this bony skull. Okay, so what happens is odorant receptor molecule shown in this colored, the red color circle, the square purple, the green triangles. So these are basically different, trying to show you the different odorant chemical structure. Okay, this chemical, odorant chemical, binds to the receptor shown in an enlarged view here. On the cell surface, on the cilia, there are 7 transmembrane domain receptor, odorant receptor, and this receptor binds to this odorant molecule. And these are GPCRs, G protein coupled receptors. So there is a G alpha, GQs, and other type of G protein coupled receptors. They will transduce the signal to the cell. Okay, so as soon as this chemical structure, odorant structure, binds to this receptor, that chemical energy is transformed into electrical activity, and then that electrical activity would travel at the other end of this neuron, so-called axon, passes through this bony skull, and then relay onto the first brain relay station, so-called olfactory bulb and the glomerulus. Okay, so individual is a glomerulus. What is special about this receptor neuron is that each receptor neuron, olfactory neuron, is specialized in expressing one and only one odorant receptor gene at a time. So for example, the receptor, for example, in the neuron that expresses the red receptor would never express the green receptor, or the green receptor neuron that would never express the red or the purple, and so on and so forth. So they are monogenic. Okay, although they have thousands of different genes to choose from, but they are monogenic. Okay, so that means one gene at a time, one gene per cell. And they are not only monogenic, but they are monoallelic. That means there are 2 alleles, one from mother, one from father, so biallele. Okay, but they expresses only from the one allele. That means they are monoallelic. So this is a unique feature of this olfactory receptor neuron. And the question that I was given in my PhD, or I chose in my PhD, is that how do these neurons become specialized? That means how do they become monogenic and monoallelic in nature in choosing the odorant receptor gene out of 1,000 available receptor gene? So one of the elegant study, and this was the key question in the entire olfactory field, not only the olfactory field, but in the field of monogenic, monoallelic expressions in case of VDR junctions and several beta globin clusters and things like that. But there was something unique about this gene family and their gene choice. Okay, so when I was just joined the lab, there was an elegant paper from the Stavros Lambordas laboratory, which suggested that, so this is the chip data. I hope you guys are familiar with the chromatin immunoprecipitations, but this panel shows here the H3K9 trimethylation chromatin immunoprecipitation. Okay, so here is a gene panel, GFP. This is MOR28 wild type. So this is one of the OR, olfactory receptor 177 OMP, which is the control gene, and then so this is a negative control gene, and this is the positive control gene, which is a repeat sequences. So in the genome there are repeat sequences, okay, repeat elements. Those are highly enriched with the H3K9 trimethylation, which is a repressive epigenetic mark. That means the gene or the area of the genome where you do see these epigenetic mark, those areas are transcriptionally silenced. Okay, they don't express. So so basically, when you hit the cells with the GFP, when you tag any MOR28 gene, one of the allele with the GFP, and when you sort the GFP positive cell with the FACS sorter, and then you try to do the chip qPCR on those sorted cells, what they observed is the cell that expresses the GFP has a very low level of H3K9 trimethylation. That means there was a selective removal of H3K9 from the allele, from the OR gene that was labeled with the GFP and that was actively expressing, transcribing, okay? Whereas MOR wild type allele, that means in a non-sorted cell, that allele was enriched with H3K9. So there was a selective removal of H3K9, okay? And not only that, in another set of experiment, they did a similar labeling of P2 gene with the GFP. They sorted the P2 GFP positive cell. That means the cell that expresses GFP, and so does they expresses P2 receptor, orderable receptor P2. And then they try to do the H3K4 trimethylation chip. So these epigenetic marks are associated with the actively transcribing gene. The area of the genome where you do see these marks are actively transcribing. That means they are firing, they are firing, and the Pol factories are active. The polymerase is firing through the gene thread. Okay, and so when you do the, when you try to probe this area at the promoter, CDS, coding region, and the 3' UTR, those areas were highly enriched. So there was a selective addition of H3K4 trimethylation and a selective removal of H3K9. Okay, so what we did in our lab is that we tried to look into the olfactory epithelia of the mouse. The dotted line represents the stem cell layer of the olfactory epithelium. Let me tell you one thing, that there are only 3 areas in the entire nervous system where the adult neurogenesis happens. That means once the neurons are born at the birth, once they die, there is no way you could replace them. Okay, so but these are the areas where there is a active neurogenesis happening even in an adult life. Okay, and in the nose, your nose gets replaced with a new neuron at every 90 days. Because there are stem cells that would divide and differentiate into immature neuron, and then those immature neuron would then replace the dead neuron, okay, the mature neuron. And so this is the basal side, this is the apical side, this is where the odorant molecules comes in a contact, and this is where the stem cell resides. So when we stained them with the LSD1, we saw a very unique pattern of LSD1 staining. Some of the cells showed a very dotted pattern, some of them were very diffuse. When we tried to do the same experiment in an in vitro experiment, what we saw is that LSD1 was a single spot, the red spot. And when we try to do the pan-OR DNA fish probe, that means it's a DNA fish that would label the entire cluster in the genome of a cell, wherever it would encounter the OR cluster. So there were several clusters, but this LSD1 was very uniquely focal plane, one focal, mutually exclusively associated with the one of the OR probe or one of the OR spot, then thus that it represents that it may be, this is where this selective de-repression of H3K9 trimethylation might be happening, because LSD1 has a two different substrate. It can demethylate at H3K9 di and monomethylations, and it can also demethylate H3K4 methylation. So it can function as either repressor or an activator. Now, in the context of odorant receptor gene, we aim to find out whether it's a repressor or activator. So this was the initial model proposed by Stavros Lambodas's lab, and this was a highly funded, very competitive lab. And some of the very foundation studies performed by this particular lab, and we follow upon their model to test their model in our model cell line. And so what their model suggests is that initially all the ORs are silenced by the virtue of having high level of H3K9 trimethylations. That means they are in a repressive environment. And somewhere along the selection pathway, LSD1 function as an activator to demethylate from a selective odorant receptor gene allele, and then that allele that is selectively chosen would transcribe at a higher level, and that higher level would then follows by the commitment or the amplifications of the transcript of the particular gene, and then that gene, the protein, would then send a negative feedback loop to downregulate the LSD1, and when the LSD1 is downregulated, the rest of the odorant receptor genes would get clustered around the highly repressive chromocenter. So chromocenters are very highly rich in H3K9 trimethylation. What you see here in blue is a DAPI, and the red is a DNA fish probe. You can see that all odorant receptor genes are either at the periphery of the chromocenter, or they are embedded within the chromocenter, and it's a kind of a jail that they would never come out to be expressed, and they remain silenced. Whereas in an RNA fish separate experiment, the express odorant receptor is pulled away from these repressive chromocenter, shown in a dark blue here, and they would reside in a euchromatic area, not an heterochromatic area, and they would transcribe at its maximum efficiency. So when we look at Stavros's data, the extensive, single cell RNA-seq data, this circle plot represents that various multiple chromosomes shown on the outer side of the circle plot. For example, chromosome 1 has this many number of genes in both heterozygous, LSD1 heterozygous animal and LSD1 knockout, completely null background, where you could see the reduction, there is a significant reduction in the number of OR. The green lines around represent the level of expression on multiple chromosomes. However, in some instances, for example, in chromosome 7, there is this gene where it doesn't follow the logic. The green bar is higher than the red bar. Okay, and so we wonder that maybe LSD1 is not a sole determinant of the selective de-repression. So we took advantage of our model cell line, which is monogenic and perhaps monoallelic in nature. And so we seeded a single cell that expresses, for example, a red receptor. This is a schematic of my assay. And we thought that in a LSD1, if LSD1 is de novo choosing the new OR, then when you expand from a single cell colony, your colony would be multicolored. That means it would express multiple different type of odorant receptor because of the gene switchings and de novo odorant gene selections. Whereas in case of LSD1 knockout, or LSD1 down-regulated cells, when you see the founder cell, if it expresses a purple receptor, then if there is no switching or there is no de novo selection, then it would commit to the same purple receptor, and the colony would become very homogeneous. And okay, and so when you take this entire colony, when you try to do the degenerate PCR, so one of the things you could do with the odor and receptor gene is that there are several conserved areas against which you could design a primer, so-called a degenerate primer, and which will try to capture as many odor and receptor gene that is present in a given colony. So we took advantage of that strategy, and we perform a degenerate PCR followed by a restriction digestion with a 4-base cutter, 3 different 4-base cutter. And the logic there was that if the colony is heterogeneous, then there are different gene products, and when you cut them with the 4-base cutter, you would see multiple bands. Okay, but if the colony is homogeneous and expresses only one odor and receptor gene, then you would see only 2 product or 2 band, okay, because there is a 1 cut only in 256 base pair, because the 4-base cutter, the product of less than 200 base pair would give you 2 band, okay, which is what we never see in a LSD1 knockdown colonies as compared to scramble control colonies. There was no apparent difference between the control versus the knockdown colonies. And so this data, and of course we cut out individual band and we sequenced and we confirmed that they were ORs. And so the primer was not completely off target or anything. And along with that data, and this data suggests that the OR activation does not seems to be dependent on the LSD1 mediated S3K9 demethylations. And then later on we perform, so this was a global assay on a multiple cells in a given colony. We performed a similar assay in a single cell where what you're looking here is a RNA fish where we have 2 different probe targeting 2 different gene. One is labeled in green, one in a red. And the idea here was if the cell is expressing 2 different gene, then 2 different colors should lit up. So what you're looking here is a single spot, which is where the transcription originates for a given gene. Okay, so we're looking single cell nucleus, and we are looking at the exact transcription origin site, okay, in the cell. So it's an unprocessed transcript, but we do see 2 different color cells, okay, and that suggests that the 2 gene can be active in LSD1 knockout background. And not only that, when in some instances you could see 2 red spot and 1 green spot, that means the allelism is also broken. So they are no longer monoallelic. The 2 allele can be expressed, and another gene could also be expressed, and we quantified the same data. And we suggested that there is no dedicated pole factories dedicated to individual gene, because we do see that the green and a red co-localization. That means they are coming from the same pole factories. All right, so we revised the model and we proposed a new model where we suggested that G9A and LSD1. So G9A is an enzyme that initially laid down or catalyzes the K9 trimethylation on various odor and receptor gene. And once you have this heterochromatic mark, they would then buried into the chromocenter and remain silenced. LSD1 functions by removing the K4 by reducing the competent OR, and it would enforce the silencing process. However, what it doesn't do is it is not a sole determinant, okay? Because the sole determinant, so LSD1 may be doing the K9 demethylations, but in a later stages, okay, right before the commitment. But rather useful function of LSD1, or one of the useful function of LSD1 prior to the selection is to keep the OR, the competing ORs silent. So that was something that we proposed, and along with these, we wanted to test if the G9A, because G9A is a major. So if we knock out G9A, can multiple OR be expressed? Okay, and that's exactly what we wanted to test. So we aim to take upon a CRISPR, which is one of the latest gene editing technology. We design and we take upon exon 2 and exon 8 of the one of the largest exome G9A. And we took on G9A, but what we found is, basically, when you hit them with the G9A, G9A has a multiple other developmental target as well. Okay, and their large T antigen was also lost. The colony became heterogeneous. And I'm gonna speed up here because we're gonna run out of time, I guess. And then we further characterize the euchromatin and heterochromatin, and the colony, the cell that was downregulated with the G9A or G9A knockout don't show the green spot. That means G9A is gone. In these knockout cells, their chromocenters are also lost. So you don't see these dark puncta of this blue DAPI, but you see one whole globe. They were diffused. When you look for S3K9 trimethylations, you do see a puncta here, but in case of G9A knockout nucleus, you see them all at the periphery. And this was a very tremendous nuclear reorganizations of a heterochromatic mark, is what we observed along with the HP1 beta, which docks onto the, which is the reader of this S3K9 trimethylation, was also lost here. And the colony becomes somewhat stem cell-like. That means the core of the colony was GFP positive, and but if you look on the periphery of the colony, there was no GFP positive cells. So somehow these epigenetically silenced, and they look somewhat differentiated. Immediately, based on my Boston Children's Hospital, I tried to look for various stem cell marker, and they indeed came positive for various germ cell layer markers and stem cell markers. Alkaline phosphatase was also positive. And so we were super excited, and those colonies on its own, without adding any supplement to it, they transform into some sort of a tertiary structure, self-organized structures. And so our lab at that time was not equipped with the stem cell lab. We were a simply molecular biology lab aimed to study the odorant receptor, but we ended up completely somewhere else. And my boss decided to write a grant on this because we were not funded to do this kind of research, and this was a completely serendipitous research. So from these, one thing I noticed is that there is a plasticity there is a plasticity and stem cell potentials. And so then that would ended up with my defense, PhD defense, and was pretty much primed to do my postdoctoral research. And I choose to do my postdoctoral research at Duke University because there was a fantastic professor there who was studying the brain neurogenesis. So in the brain, there are only two areas where the adult neurogenesis happens. One is in the subventricular zone, and the other is in the hippocampus, associated with the learning and memory, and subventricular zone associated with the formation of the entire cortex at the embryonic and the postnatal area, postnatal age. So this is my last part where I would talk about identification, characterization, and activation of quiescent neuronal stem cell. And this was a transition from Wesleyan University to the Duke University on the left. And as my innate drive, I always wanted to go from a nose neurogenesis to the deep into the brain neurogenesis. And so this was a very nice transitions, and I found this lab to be very interesting. Also, when you compare the olfactory epithelial neurogenesis, their structure and the function were somewhat very similar. So what you have here in the nose is basically when you take the coronal sections of the mouse nose, when you flip it, all these villi has this kind of a structure where you have a multiciliated sustentacular cell, first layer, and then followed by this yellow cell, which is a mature odorant receptor, followed by immature orange cell, And then the stem cells, both globose basal cells and the violet horizontal basal quiescent stem cell reside in this niche. Okay, and then this yellow neuron gets replaced at every 90 days in the nose. Whereas if you look at the brain, when you take the coronal sections right from the middle of the brain, and when you flip it towards you, this is what you see, where you have corpus callosum shown in a white here, okay, and above the corpus callosum is all cortex area, and below the corpus callosum there is a dark pink color here, and there is a lateral ventricle, okay, and there is a lateral wall. The wall that supports the ventricle is called a lateral wall. Now, if you look into this cartoon at the bottom part, their anatomy is somewhat similar to the nose anatomy, and where you have a multiciliated ependymal cells, and followed by these neuronal progenitor cells, neuronal stem cells shown in a green. They would divide into these neuronal progenitor orange cells, transit amplifying cells, and then they would further differentiate into these neuroblast red. Those neuroblasts would migrate along the rostral migratory stream, and they would come all the way at the olfactory bulb to differentiate into different granular cell layers, perigranular cell layers. Because I'm always curious in my PhD that there is information coming in, the nose is receptive to these informations. How is these informations gets processed into the brain? Okay, there has to be some compensatory neurogenesis coming to the nose, to the bulb, which would then somehow coordinate to the information coming through the environment and interpreted by the brain. Okay, so this was a fantastic idea to go from the nose to now understand the OB neurogenesis and SVG neurogenesis. So I'm gonna skip this and jump directly into this talk here. So basically, we wanted to use these SVG neurogenesis. So this is a cartoon showing the coronal section of the brain where you have a lateral ventricle shown in a watercolor, and then followed by the pink wall is SVG, subventricular zone. This is where the stem cell resides. Whenever there is an injury in the brain cortex, this stem cell responds to the injury. Okay, this is the ipsilateral side, which is where the injury happens. It's a contralateral side, which is where the injury is not happen. Okay, so the cells would not migrate to the contralateral, but they would migrate to the injury sides. So one of the key question is, and as you can see here, that this is N4 RTM cells 3-day post-injury in the left panel. This is a 14-day post-injury in the right panel. And you can see that TD tomato, N4 RTM, N4 is a neuronal stem cell marker, molecular marker. They are labeled with the RTM TD tomato. And when there is an injury at 3 days, they don't migrate. But at 14 days, you do see this long stretch of cells migrating from lateral ventricle, going all the way, crossing this corpus callosum, going all the way to injury site. Okay, and what do they do there once they go to the injury? So they differentiate into these GFAP positive cells. And one of the key thing there was, okay, so the thrombospondin 4 was identified already as a stem cell marker. And it was proposed that it is the T4 thrombospondin 4 positive cells are the one they're going to make differentiate into neuroblast. And so here you can see the thrombospondin 4 positive cells, all TD tomato positive cells are thrombospondin 4 positive. Thrombospondin 4 on the ipsilateral side was higher signal, and you can look at these TD tomato positive cells are pretty much positive for thrombospondin 4. So then we went after thrombospondin 4, and we wanted to look at the behavior, selective marker for quiescent neuronal stem cells. So this is the genetic strategy that we developed for the mouse. And if you look, so what you do is basically, in the animal, at P28, postnatal day 28, when you inject with the tamoxifen, so tamoxifen would activate this Cre, it would bind to the Cre and it would activate the Cre recombinase to activate the TD tomato, the ZS green in this case. And when you look into the olfactory bulb, you don't see any ZS green positive cell 1 week post tamoxifen injections. But when you do see, when you wait 3 weeks post tamoxifen injection, you start to see the cells migrating from the SVZ to the bulb. So this is the olfactory bulb, and this is the complete bulb, where you can see that there is penetration into all different layers of the olfactory bulb. That means they're contributing to all the layers, all the 6 layers of the olfactory bulb. Okay. And when we co-stain them with the MASH1, some of the cells were positive for MASH1 too. That means MASH1 is a pro-neurogenic transcription factor. And so there was it confirms that they have an ability to differentiate into neuron. And when we pulse them with the EDU at the 3 week, as compared to 1 week, the 3 week was higher. They took a higher EDU as compared to the first week. That means there was higher proliferation at 3 week. And so how do these quiescent neuronal stem cells gets activated? Okay, so what you do is in the SVZ there is already a canonical stem cell that proliferates. So you have to kill those cells before you try to understand whether there is any quiescent neuronal stem cell that has the potential to be activated. So what we did is we treat them with the Araxi. Araxi will kill any proliferating cells. Okay. And then, we treated them for 5 days and we analyzed at day 0. And then we waited another 1 week and then we reanalyzed them. Okay. So what you see here is a ZS green DCX panel, and this is the merge panel. At day 0 panel and day 7 panel post Araxi treatment, you can see that day 0, you don't have any DCX positive cell, okay? Whereas when you wait day 7, there were DCX positive cells. That means these cells must be coming from the ZS green positive cell, and they were indeed ZS green positive. So you can see here, this is ZS green with co-labeling with the DCX, okay? So all the canonical neuronal stem cells became dead, and whatever the new cells were generated, neuroblasts were generated, were all coming from the ZS green positive activated quiescent neuronal stem cell. So we took this assay into in vitro. So we were looking for what it is that activates the quiescent neuronal stem cell. It turned out that there is a ependymal cell secreted noggin that activates the quiescent neuronal stem cells. So when you treat them with the noggin culture, you see higher level of DCX positive cell as compared to control, no treatment control. Okay. And so when you treat them with the shRNA in the brain against the noggin, you should expect the reduction in the neuroblast production, which is what we observed. So here is a control DCX cells, higher number of positive DCX positive cells. When you treat them with the noggin shRNA, you lose the DCX positive cell expression. That confirms that when you add, there is a more productions. When you take the noggin out, there is a less production. So noggin, which is the BMP inhibitor, is one of the activator of quiescent neuronal stem cells. Then, so we identify T4 as a quiescent neuronal stem cell. We now know how they can be activated. Now, the third question was, if they are a true stem cell, can they contribute to various different lineages, neuronal lineages? So what we did is we perform a single cell sequencing, and I'm not going to show you that data. But what I can show you right now is that they were GFAP positive. Okay, so there was a glial production. They were FOXG1 positive biciliated cells. That means there was a other cell type that resides in the niche, they were positive. And not only that, those ZS green positive cells were acetylated tubulin positive. That means they were functional ependymal cells. Okay, so they were multipotent in short. We wanted to see if these ZS green positive cells can contribute at the stroke site. So what we did is we injected tamoxifen P17, waited for 10 days. We induced the cortical stroke, photothrombotic stroke, and then we perform the analysis after 16 days. Okay, so this is the injury site. This is where the injury happened, where there's a black hole. Basically, multiple different cell types die, because you are inducing the photothrombotic stroke. So blood vessels die, glia die, oligodendrocytes die, neurons die, and they would never regenerate unless if you provide supplement from the outside, if you do the transplantation of stem cells from outside, or you induce the endogenous stem cells to migrate there and differentiate into various different cell types. So what you're looking here is ZS green positive cells can reach to the injury site from the SVZ, and they differentiate only into the GFAP positive cells. We never see that they are differentiating into DCX positive cells. Okay, so this is a major challenge in the field right now, that the SVZ is responding to the injury, the quiescent stem cells are going to the injury sites, but somehow they are differentiating into GFAP positive glial cells, but they are unable to differentiate into neurons. So once there is a neuronal deficit, there is no current therapy by which you could replace the neuron, and not only replace, but those new neuron can integrate into an existing circuitry. We try to look into the human brain, and I can tell you, this is the very latest data, that in the birth, there are a lot of Ks67. I'm gonna wrap up in 2 minutes, okay? Maybe I have 1 minute. Okay, so, all right, so there is at the birth, okay, this is the birth, there are higher Ki67 positive cells, and there is no or almost low level of thrombospondin positive cells. So that means at least at birth, all the cells are engaged, neuronal stem cells are engaged in dividing and producing a new neuron, okay? And there is no quiescent neuronal stem cell. But as the human age, 7 months, 2 months, 7 years, 30 years, 55 years, those potential of proliferation of neuronal stem cell is reducing. You see the green dot is reducing. Ks67 is a proliferation marker, and thrombospondin 4 shown in the red is increasing. That means majority of the cells are becoming quiescent. So at the age, once there is a neuronal deficit, human brain perhaps already has a quiescent neuronal stem cells, but somehow if you can develop some therapy or treatment, some way to activate these T4 positive cells, then maybe those neuronal deficits can be well taken care of. And so we look deep into the brain, and so these T4 positive, thrombospondin 4 positive cells, we try to map them at where exactly they resides when the human age. They reside at some of the temporal lobe and some of the dorsal side of the lateral ventricle at age 60 years and age 37 year. And at birth. At birth, you don't see any T4 positive, the red staining. But as the human age, you start to see a lot of red foci, and at 60 years, pretty much all red. so they become quiescent, and this is just a RNA fish data. I mean, it's a fish probe, and as you can see here, this is a quantification of the same data, that T4 GFAP positive cells are higher at all ages, okay, or at all ages as compared to the negative control probe. Okay, so This is where that story stands. Now going from there to my future directions, one thing we do know that when there is an injury, there is a cell migrations. How do these cells migrate? How do these cells that are residing here sense that there is an injury at the very distal part of the brain? So it turns out that the neuroblast binds to these blood vessels shown in a purple, and along the blood vessels is what they migrate. But when there is injury, everything dies, including the blood vessels, microvessels, everything dies. So if there is no blood vessel, there is no direction for these neuroblasts to migrate. So currently I'm working on angiogenesis into this infract area, and hope perhaps with the enhanced angiogenesis, we will have a more facilitated migration of these neuroblasts, and we will be able to see more DCX positive neuron there. And I'm trying to develop the therapy here. I'm not going to go into detail here to bore you guys, but this is basically showing you that there are certain areas which can be targetable. So this is the core of the injury, which is a black hole. There is nothing surviving here, oxidative damage. But around this core area, there is a penumbra area, peripheral area, where there are some blood endothelial cells are surviving. We can make them proliferate faster, and we can regenerate the angiogenic microvessels to which these exons can grab on for the nutrition and other supplies, and they survive. And as soon as the new neuroblast would come from the SVZ, they would differentiate and repenetrate and reorganize, make the network of neuronal connection again. The circuitry will be re-established. All right, so with that, I would like to thank Dr. Dhruva Sen from Techno India University to give me this opportunity to be with you guys today. And he's been an excellent mentor throughout my life. And I'm very thankful to him. My PhD mentor, Dr. R.P. Lane, my postdoctoral advisor, Dr. Che T. Ko, my research assistant advisor, Dr. David Williams at Boston Children's Hospital, and my current postdoctoral advisor, Dr. Rajkumar Verma, for my angiogenesis and neurogenesis project, and their funding agencies, National Institute of Health and National Science Foundation at the USA. And these are my committee members and former collaborators. This is just an epigenetic landscape that opportunities will fall. Okay, so once you are stem cells, you are multipotent. You're gonna go downhill, take one path as a student, but always remember that you could always be reprogrammable. Okay, so I was in a blood field, then I went to cardiovascular field, genetic fields, neuroscience, angiogenesis now, so endothelial, and I'm always changing and reprogramming myself, trying to acquire the knowledge and apply the knowledge for the best of the science. Thank you very much.
Thank you, sir. Thank you very much for your session. I'd like to now call Dr. Dhruva Sen, sir. Hello, sir. Are you there? Hello. Sir, are you there, sir?
Dr. Shen, are you there?
Okay, I am here.
Okay,
I am
sir.
here,
Okay,
here. Many thanks, Dr. Vyas, for your nice talk and nice speech.
Thank you.
After 15 years, we are listening you, especially me.
Yeah. Eddie, I would be happy to take any questions if anybody has any.
Is there any questions from anybody or from audience?
No, sir. There is no questions yet, sir.
Okay, thank you, Dr. Vyas, for your nice and excellent speech on stem cell research.
Okay, so it's 12:36 a.m. here. I'm unable to continue this webinar session I would love to hear them, but I think there is a recording going on. So if possible, can somebody send me this entire excellent webinar series? And I would
Okay, no
be
problem.
to listen at my convenience, but it's actually heading towards 1 a.m. here right now.
Okay.
It's, okay, so good luck to all of you. Take care.
Thank you, sir.
So, sir, thank you for the session. Wonderful talk. It was really a wonderful talk.
Okay, take care. Bye-bye.
In advance, sir, Merry Christmas.
Merry Christmas to you.
You-
Merry Christmas to all of you.
You
Yeah?
just right now you are in USA, so you just enjoy your winter vacation. Okay. So good luck,
Yep.
sir.
Yep, wonderful. Thank you very much. And Merry Christmas to all of you guys. Happy New Year in advance. Okay, take care.
Okay.
Okay, okay.
Thank you so much.
Thank you,
Yeah.
thank you.
Thank you so much, sir. Thank you. Bye, sir.
Okay, bye. Now I am happy to introduce our last and fifth speaker. Supradeep,
Yes, sir.
Supradeep, are you there?
So our last and final speaker. Yes. Am I audible, sir?
Yes, you introduce first.
Okay.
Hmm.
So I'd like to introduce our last and final speaker of our today's webinar. So we're in the final day. Dr. Dhananjay Das is with us right now. He's the final speaker or last speaker of the day. Dr. Shaha, Dhananjay Shaha, he completed his B.Pharm from Jadavpur University. Then he joined the masters from the same university and did masters in biomedical engineering. Then he did his PhD from Jadavpur University. He's having huge research experience, about 20 years almost. His area of interest for research work related to the intellectual property right, pharmaceutical and biomedical engineering, tissue engineering, and sports science. He has also worked as an examiner of patents and design. He has also worked as assistant controller of patents and design, intellectual property office, Ministry of Commerce and Industry, Government of India. He is the life member of Society of Biomaterials and Artificial Organs. He is also associated with Indian Institute of Chemical Engineers, the Institution of Engineers. He has published more than 50 different scientific papers in international and national journals. Right now, he's working as the Deputy Director of Technical Education, Directorate of Technical Education, Government of West Bengal. So it's my privilege to welcome you, sir, in the final session. Dr. Dhananjay Shah, sir, are you there? Can you hear me? Can you hear me, sir?
Dhananjay, are you there? Hello? Dhananjay?
Sir, I can go through the names of Dr. Shah in the panelist name. So there may be some kind of technical issue, I guess.
Are you there?
Dhananjay sir is there. I think there is some network issue.
Yes, yes, yes.
I think sir is facing some network issues, sir.
Hmm.
Hello, are you there? Yes. Dr. Dhananjay Shah is our last and final speaker. Dhananjay, are you there?
Yes, I am here. Am I audible?
Yes, yes. You are completely audible. So let
Yes,
me
sir.
introduce.
You can
So
start your
today,
session.
okay, sure.
Okay.
Okay,
So
should
let
I
me.
start?
Hello. Let me introduce
Yes,
Dr.
sir.
Dhananjay
Hello.
Saha. Dhananjay
Yes.
Saha is my friend of B Farm and best friend. Dhananjay means Arjun of Mahabharata. And another name of Dhananjay is winner of wealth. Winner of wealth is Dhananjay Saha. He did B Farm with me from Jadavpur University, and the batch is 1992, the B Farm batch. Then he did Masters of Biomedical Engineering from Jadavpur University and PhD from Jadavpur University. And his most profile has been read by Supradeep. The remaining part I am discussing. He has published more than 25 papers in international journals of high impact factor with me. After joining in Techno India University, I have published more than 25 papers in international journals and filed 2 patents. And recently, we are going to publish a book that is Foundation of Body Homeostasis Salutes Coronation of Engineering. Publisher is Degenerate Publishers, Australia, and the authors are myself, Dhananjay Saha, and Kushal Nandi of third year B Farm. Kushal Nandi has published so many papers, at least 20 papers, me at least I think so, 20 papers with Dhananjay Saha and myself in very national, international journals of high impact factor. And I think he will continue afterwards also. So Dhananjay Saha is expert in IPR, patent, intellectual property rights. And he has so many expertise in IPR, and he will deliver today's lecture on topic is application of intellectual property rights in pharmaceutical and biotechnology research. So he will compile pharmaceutical research and biotechnological research is in IPR today. So Dr. Saha, you please continue. Dhananjay, are you there?
Dhananjay sir, please unmute. Dhananjay sir, please unmute.
Dhruva sir, are you there? Dhruva sir, are you there?
I am here.
Okay, sir. It's a request to you. If it is possible, then tell Dr. Shah to leave the session and then again join, because what I guess he's facing tremendous problem with audio and video. Some sort of internet issue may be there with, or
But I
some
am trying,
problem
but call
related
is not
to the
going.
device. Okay.
I am trying, but call is not going. হ্যাঁ হ্যালো, হ্যাঁ বলেছি ডিসকানেক্ট করে নতুন করে জয়েন করতে।
ওকে স্যার, ওকে স্যার, ঠিক আছে। স্যারকে বলুন নতুন করে জয়েন করতে, নেটওয়ার্ক ইস্যু হচ্ছে। আর স্যার বলুন ভিডিওটা যেন অফ করে কথা বলেন উনি।
হ্যাঁ বলো। ওকে ঠিক আছে, কন্টিনিউ। হুম। Please wait. Dr. Shah is connecting.
হ্যালো সুপ্রদীপ স্যার, হ্যালো।
Yes.
হ্যাঁ স্যার, বলছি স্যার ধনঞ্জয় স্যার তো এখনো জয়েন হতে পারছেন না বোধহয়, একবার দেখুন না।
হুম।
হ্যাঁ,
না,
দেখুন।
আমি ওটাই লিখছিলাম, আমি ধ্রুবজ্যোতি স্যারের সঙ্গে আমি কথা বলছি।
ওকে
হ্যাভ
স্যার,
পেশেন্স, আমি দেখছি। হুম।
ওকে স্যার।
Dhruva sir, are you there? Can you hear me?
Yes, I am here.
Okay.
I have talked with
Uh,
Dhananjay. He has some problem, technical problem, and he is trying to connect.
okay.
Okay, no problem. You
Okay.
please wait.
Thank you, sir.
Hmm.
আছে?
Dr. Shah, can you hear me?
I am audible.
Sir, your voice is cracking.
Okay. चलो बाहर. Okay, just wait.
Now, sir, you are completely audible.
Audible now?
Yes, sir.
Okay, fine. Actually, I'm very sorry for technical problem. Okay,
No,
now
no
it is
problem,
clear.
sir, no problem. Yes, sir, absolutely. You can start your session right now.
Okay. Okay. Fine, Rushal. Am I now?
Yes, sir.
Hello?
Clearly audible. Please continue.
Okay, first I am extremely very sorry for some problem. First I started using my PC, but some problem is there. Not necessary. So again I did my lecture by using my lead assistant. Okay, Omshul Shah. Okay, very good. Now it is still morning or not going to noon. Okay, good morning to all. Professor and best friend, Dr. Drupajyoti Sen, and Professor Director, School of Pharmacy, Techno India University. Good morning to Kushal, Ogo, Pritam, and Suprati, Olunvo, Shandip, Pamani, and all the participants.
Good morning, sir.
Am I audible? Hello?
Sir, your voice is cracking.
Tracking. Now it is okay.
No, sir, your voice is not clear, sir.
Hello? Okay, now is audible. I am
Not completely, sir. Still now there is some there are some issues with audio.
with audio. Okay. I think is it clear?
Yes, sir. Yes, sir.
Is
Completely.
it clear?
Yes, sir.
Yes, sir. Now it is clear.
All of the research group are very done lot of activity the guidance of Professor Dhruva Sen. I discussed with Professor Sen. We shall go ahead next year, 2021, in a fresh mood and try to get some funding projects so that we can go further ahead for innovative idea and complete in form publication, and it may be implemented and submit to patents for research output. So now I'm happy opportunities in
Sir, your voice is fluctuated.
is it clear? Now, am I audible?
Yes, sir. Yes, sir. You are audible.
Okay, fine. So big problem. So why you should learn, students, IPR, intellectual property rights. Intellectual property right deals with so many things, especially students, those who are researcher in future would be researcher. Then IPR knowledge is necessary. I said, so student and area of what different academic. Chemistry, another biology. Chemistry means so many problem.
Sir, you are not audible perfectly.
Hello?
You are not.
Hello, you are not
perfectly-
audible.
Hello. Hello. Now audible. Am
Yes,
I
sir.
audible?
Yes, sir.
Okay.
Yes, sir, you are audible.
Okay. Okay.
Yes, sir.
So what I am telling students, why need IPR, and if you see pharmacy, it's a interdisciplinary area. You can do work in different versatile area. It depends on your mindset and your compatibility. If
তাও তো শোনা যাচ্ছে না। হ্যাঁ, তো এরা তো স্ক্রিনশট করে দিচ্ছে, শুধু রিড করে যা।
ওকে, তাহলে আমি তাই করে যাই, স্ক্রিন ওটাকে চেঞ্জ করতে বল। ওকে, নাও আই এম go to next slide. Go to next slide. Yes, you see Albert Einstein, after completion his PhD from University of Zurich, he joined patent office as a patent examiner, and he said his patent IPR knowledge help for further his activities. Next slide. Next slide. Am I audible? Hearing?
Yes. Yes, audible.
Yes. Next slide. Audible. So okay, students, I am telling what is invention versus discovery. You see, discovery means which is exist in the nature or in the throughout the world, or in the this is discovery. Invention is not a existing in the nature, it is invented by someone. So in case of patents, discovery is not a patentable subject matter, only invention is patentable. Basic things. Next slide. Okay, difference between invention and innovation. See, invention of wheel, it's a big invention, and by using wheel, it we are producing car. So this is the difference between invention and innovation. Invention means produce some new things, and application of these new things in commercial or any other use called innovation. Similarly, World Wide Web is a internet is a invention, and using that web is a innovation. Next slide. Okay, so if we see our previous, our old heritage and long back, what was our thought? Our thought says ancient knowledge should be freely shared. But it is our thought, and previously we believed that. But you say knowledge is not free, it is at all. So knowledge means your knowledge you can apply for invention, you can get a use that invention, or in intellectual form you get some benefit, and I'm coming later. Next slide. Okay, intellectual property, what is it is called property? You see, property means house, money, etc., gold, all are properties. But intellectual is one type of property. Intellectual property has acquired the term property because the owner of the right has legal power to lease, sell, donate, license, etc., with similar status property. What are the different parts of intellectual property? You see, first is patent, second is design, third trademarks, copyright, geographical indication. Patents, basically, what is patent? Patent basically your invention. Design I mean not engineering design. Design means aesthetic design. If you develop certain aesthetic design of a car or of a vessel, like so many things. Trademarks is also a part of intellectual properties. Copyrights. Copyrights is a part of intellectual property right. Copyright means software. Yes, software under the copyright, and books all are publications. Books all are copyrights. So everybody knows because you are also doing publications. So it is under, you have to submit the copyright forms. So geographical indication. What is it? Is GI. GI means geographical indication means suppose Darjeeling tea, Baluchuri Shadi. These are geographical indication. This invention or this knowledge in particular area. So it is under GI in geographical indication. Basically, we shall discuss here only patents because we are going to further research. So your invention, your research, how to patent it, how to do it, what is not necessary, I will discuss these details. Next slide. Okay. What is patent? So friends, I will take, it may be exceed 12 o'clock. It may be little bit time because it was some problem to start. I already 30 minutes west, I think. So okay, I request bear with me. Am I audible? Hello? Hello?
Yes, sir. Yes, sir. You are perfectly audible.
Okay, thank you. So how much time I can continue? If first tell me, because already I late. So everybody will take lunch. So it is depends. So you fix the time.
Sir, you
Sir,
can continue, sir.
Yes, sir. Your session
We
is
have
the
no
last.
problem with it.
Okay, thank you.
Okay.
So patent. A patent is a monopoly right granted to an inventor, giving the inventor the right for a limited period of time or prevent others from making, selling, using his invention without due permission from him.
Okay. হ্যাঁ 20 মিনিটে শেষ করো।
Okay, share. Okay. Cancel. Share cancel. Okay, then patents. So it is for a particular period of time. If suppose you file a patent, you get the patent. If it is granted, obviously. So it is time periods. That period you can enjoy the monopoly right. What is the time period? If you file the patent, so if it is granted after examination of all the procedures, it time period 20 years. Throughout the world, this is the same rule. After getting the patent, granting the patent, is 20 years your monopoly right. So you can use the patent, commercialize the patent, and you can earn the revenue and of the patent without your assignment. Nobody can use that patent. Okay. So patent can be bought, sold, rented, hired, etc. Next slide. So infringement. So if anybody without your prior permission using that patent and commercialize the patent, so you can protect it, you can you can take action against that infringement. This is the because patent is your legal your only your right for certain times. Next slide. What this means to you? What is the use of patents? Why you are doing patent, and what is the benefit of the nation of the patent? What is the benefit of the inventor of for filing a patent? So many things. So first thing, promote invention and help safeguard your inventions. Promote inventions. You see, in pharmaceutical industries, basically those are doing research and finding say new molecules, product or process, anything. It's a huge investment. Use lots of investment. Even you see R&D pharma companies investment. If you compare to 5 10 companies, it is the only pharma company, R&D investment is lots of money. So why companies are invest for invention? Obviously, promote invention, and patent filing helps to promote invention and help safeguard your invention, means protection your invention, help companies grow, benefit the community by making new goods and services, obviously, because promote the invention, safeguard the patents. So obviously new molecules, new products, new inventions coming up. Provide personal growth, development, and advancement. Obviously, inventor will get the benefit from the for his invention by getting patent. Next slide. So these are the objectives for filing patents in any area. It may be pharmaceutical, it may be product, it may be process. So what are the objectives? Encourage inventions. Inventions become known instead of remaining secret. Industrial development, wealth of technical information. Next slide. Who can file a patent? Anyone who contributes to the conception of an invention alone or in combination with others. Somebody developed a process, pharmaceutical process development process, or somebody, if you any researcher in laboratory, somebody developed a molecule, say maybe anti-cancer, anti-tubercular, any drug. So inventor individually with this researcher individually or group of inventors can submit the patent for granting of the patents. Okay, next slide. Please, next slide. Invention basic criteria. What should be the basic criteria for granting a patent? First thing, it should be novel. What your work you have done, you developed a molecule, you developed a process, you developed a product, or anything. First, it should be novel, means it should be new, means it is not published. Your product or your molecule or your process, you're not published anywhere, means any public journal or any patent document. So it should be novel, basic criteria. When you're going to submit the patent, whether it is novel, basic, you satisfy it, basic criteria will fulfill. Next, non-obvious. What is non-obvious? That is the difference between your invention and prior art that would have been obvious to some in the field. Means if it is exist in the prior art, so it is not obvious. Should not be in prior art. This should be non-obvious. Second thing is, first thing, second, non-obvious. Next slide. Right. What is prior art? Prior art means so basically students for third year pharmaceuticals, you are doing so many expert in research, doing publication, and I talking with regarding you with Professor Shane is really excellent. They are doing sorts of review articles, doing that. So really I surprised because you are undergraduate. You are doing so much idea regarding how to research. It's a good thing. This is the first time I told so in Professor Chen. So this is very good. You are the only person. You are doing. You are trying to learn how to do research for the students. Okay, fine. So prior art. So prior art means when when we going to publish a patent or printed, it should not be prior document, means that document should not be published anywhere. Prior art includes patent, printed publication, other disclosures in the field of your invention that have been published before your effective filing. That have been published before your effective filing date. Okay, next slide. So patents, what are the things you can file a patent? First thing, okay, I am brief going. In 1995, GATT agreements had been happened GATT and TRIPS agreement. Before GATT and TRIPS agreement, only India, in case of India, in case of pharmaceuticals and agrochemicals, product patent only acceptable. Means, process patent was only acceptable. No product patent was not allowable as per Patent Act. Or so process patent means, so you developed a process, different process by producing the same molecule, that process is patentable. But product, suppose you developed a new molecule that was not allowable before GATT and TRIPS agreement. After GATT and TRIPS agreement, India also allowable to file a patent in product patent in case of pharmaceutical agrochemicals. It was implemented from 2005, January 1st January. Product patent was implemented in Indian patent system. So especially product means, again I'm telling pharmaceuticals, but product, any product, say device, say computer components or anything product was also patentable in that time period. Also, the only changes in patent allowable after 2005 January. So patent may be process, product, composition matter, and so many things. So you can file. Next slide. So again, I'm telling discovery versus invention. Discovery is not patentable subject matter. Invention only patentable subject matter. But in case of, you see, sometime discovery may be converted into invention, then it is patentable. How? Suppose discovery means finding a new substance in nature, elucidation of the chemical structure of the natural product. Suppose you find out a plant, that plant's fruits or leaves contains such a type of alkaloids. Okay, that's alkaloid, that plant exists in nature, so it is discovery. But if you isolate the plant, if you isolate the particular alkaloid, and if you can show the efficacy of the alkaloid, structural elucidation you have done, you have explained the efficacy and pharmaceutical action of that compound, then discovery may be converted in invention. So invention means creative application of scientific knowledge on technical field, requires additional step towards a technical solution problem used for public. Discovery can be basis for invention. Next slide. Yes. Suppose you isolation of the substance, structure elucidation of the substance, maybe say alkaloid collected from the nature, and use of this substance as a drug, particular alkaloid, then the discovery finally turns into invention. Next slide. You see some example. This is a fungus. It is green death carp. So isolate this structure, compound, antamonite. It is used as a antidote against poisoning. So you can say this compound is existing this fungus, so it is discovery. No, something here concept is different, because you have some intellectual input in this work. You isolate the compound, you have studied the activity of this compound, you next. Okay, next. Right. So basic criteria to be patent is novel, inventive step, and industrial application. First time, novelty. You see, suppose somebody this paragraph means already this method, this molecule is published in paragraph. A medicine for diseases comprised of. So it is not obviously novel. Next, a medicine for diseases comprising effective component B. So you develop compound B, different compound, but disease is same. So new molecule, the same disease, so it is a novel. But if you see a effective compound, compound A, but disease is different. So new use of known substance means disease is had effective component A, but it is used in X different disease, Z disease. It depends. It's obviously invention depends upon the patent act. What depends upon the patent act is the territorial right. If you go see the Indian patent act is different. If you see the European patent act is different. If you see American patent act is different. So almost axiom act is same, slightly different. That is, some portion is there, varies to country. If you see the Indian Patent Act, so new use of known substance means suppose to you know aspirin as an analgesic or use as a blood as a heart disease. So you find a new aspirin use also, suppose anti-cancer. You find out aspirin also used as anti-cancer. It is an invention, but aspirin is non-compound. Finding use is different. As per Indian Patent Act, it is not patentable. But if you compare to other countries, US, if you file the patent in USA, it is allowable. Next slide. Inventive step. So first criteria to get a patent is novelty. Then inventive step. Inventive file a patent. And the claim part of the specification is written.
Sir, your voice is rapturing.
Capturing. Is it audible?
Yes, sir. Audible.
Now it is audible.
Okay. Audible. Okay. So inventive step, okay. So if what paradigm says the particular drug comprising compound A and B, and result of pharmaceutical performance of the synergistic anti-cancer effect. And prior art says it is known that compound B and compound A effectively have anti-cancer effect. So adding of this compound B causes anti-cancer effect. By adding two compounds, same anti-cancer will get the synergistic effect. By adding this compound, then it may be patented. But means it's by the inventive step. Okay, next slide. Next slide. Very interesting thing I am telling. So, in case of microorganism, patentable substance of microorganism, whether microorganism is patentable or not, you see, you can say microorganism is existing in nature, so it is not patentable because it is discovered. First man, Professor Anandamohan Chakraborty in the world, recently last 2 months back, he died. He was in professor in University of Illinois. So what he did when he was in after completion PhD from Calcutta University, he went USA. First he joined GE, General Electric Company, and he developed a microorganism. It is one type of microorganism. It is genetically microorganism. It is using for using destroying the oil. It means pollution. Remedy of the pollution, of the oil pollution. So he filed that microorganism, the Pseudomonas, and in US patent office, G company filed the patented that microorganism. It is useful microorganism for prevention of pollution. So oil pollution in sea level. So, and what if US examiner told, US patent office told, no, it is not patentable because it is not invention, it is discovery. Microorganism is in nature. It's a famous case in throughout the world. This is the first case, Diamond versus Chakravarti. Court case, famous court case. Ultimately, he is the first man in favor of him in G company granted the patent. But ultimately, what they told it's not a this organism, microorganism previously was not exist in nature. After recombinant DNA technology, we developed that microorganism, and then that microorganism uses some enzymes. That enzyme lyses the oil, breakdown of oil. So this is not discovery, this is invention. Ultimately, that patent was granted, and he is the first man. Microorganism patent was granted in USA. And if you see in India also microorganism is patentable at present in past Indian Patent Act. But if you go on higher than microorganisms, means I see transgenic level, transgenic mouse. You know, transgenic mouse we are using for research of anti-cancer drug. So transgenic mouse already patented in USA, and that patent period already over 20 years over. So, but if you see Indian Patent Act up to microorganism is patentable, but higher than microorganism or transgenic level is not patentable. It's slightly different as per Indian Patent Act and US Patent Acts. Next slide. Next slide, please. Okay, Indian Patent Act. If you see Indian Patent Act, it was 1970, and so many amendments. 99, 2003, 2000. New Patent Act was implemented in India 1st January 2005, especially for coded patent or after that and these agreements. Coded means pharmaceutical molecules or agrochemical molecules. What I'm brief in few examples, which is not innovation. Is it
আবার এসছে।
Am I audible?
Yes, sir, you are audible. Yes,
Okay,
sir, you are audible.
sorry, I am not. Sorry, I am not really surprised. Actually, this is the first time. This is I facing problem. So maybe some networking in my.
Okay, sir.
Sorry
That's
for
not
that.
the problem. Okay, sir.
And so I am giving some example which is not patentable. Suppose anything related to atomic energy. This is not patentable. Anything related to human moral and against the ethics. Suppose you developed a product by using the human embryo, is it not patentable? So so many acts, what is patentable and what is not patentable. Next slide, go to. Gonava, please, next slide. Okay. Non-patentable, yes. What is not pharmaceutical patents? Method of adulteration, food, producing brown sugar. Next slide. Okay, method of treatment, very important thing. In India, patent may be, however, often a form of administrative drug is not patentable. Suppose you the doctor developed a surgical method. Suppose new method of bypass surgery or any surgery. It's a method of treatment. Even it satisfies the novelty, it satisfies the inventive state, but method of treatment, it against the Act, Indian Patent Act. But few countries are allowing method of treatment is patentable subject matter. Next slide.
Sir, you are not audible. Sir, can I go to next slide?
Yes. Okay, turmeric, yes. Turmeric is a big history, I'm telling to you. Turmeric, yes, wound healing. So he filed the audible? Am I audible?
Yes, sir. Now
Hello?
you are audible. Yes, sir. Yes, you are audible now.
Turmeric, what has been happened, US scientists filed a patent in US patent office, for patent for it used for a wound healing, and he finally got the patent. But Indian CSRs filed against that for the revocation of the patent. What is that? Because turmeric used as wound healing our traditional knowledge. Because we have so evidence, we produce that US patent office, yes, turmeric is a new wound healing. It is already long back, is a traditional knowledge of in India. So submit the document, ultimately revocation of the patent, withdraw of the patent has been done. It's a big case fighting this CSIR versus US patent office. Next slide. This is the issue of patent of turmeric. This is a certificate issued by, this is the patent number issued by who awarded University of Mississippi. medical center in 1995. After fighting, CSIR purchased US Patent Office, and ultimately that patent was rejected. Next slide. You see, in India, while the turmeric has been medicinally for thousands of years, grew about for economically. CSIR argues that turmeric has been used for thousands of years for healing wounds and rashes. Their claim was supported by the documentary evidence of traditional knowledge, including ancient Sanskrit texts and paper published in 1953 in the Journal of Indian Medical Association. Next slide. This is the case. US patent granted in turmeric 1995 for examination at USPTO. USPTO means United States Patent Trademarks Office, October 1990, and ultimately a re-examination certificate issued and proceedings concluded in favor of CSIR. Next slide. So, development of the country. If you see the number of filings, how much filings in patents in every country, you can understand the development of the country. Means, if suppose India, what are the filings? Some indigenous filing and some patent filing from the abroad. Because patent rights is a territorial right. If you Glaxo file a patent in USA, in Glaxo monopoly right only for USA. So what Glaxo trying to file in the patent in so many countries. So he will patent rights from other countries also. So what is happening in India? If you see the scenario of India, you see total 100%, 90% filing are coming from the abroad. An Indian patent office for granting patent, only 10% Indian researcher filing individual or CSR, some filing are doing. So if you see, in case of India, indigenous filing less than 10%, but in domestic filing, indigenous domestic filing less than 10%, and more filing from abroad. In this developed country, domestic is more equal to the why patents? Because patents, it's a for pharmaceutical. If you see, R&D investment is huge for getting a molecule. You all are pharmacists, you know how much step is required for granting for commercialization of a molecule. You see, first you have to find out the molecule, then go for clinical trials, or for phase 2 trial, phase 3 trial, then go for commercialization. So there are lots of investment, lots of happening. So even after phase failure. So if some company gets new molecule, so he is trying to get a patent because he can enjoy for a particular period monopoly rights to recover revenue. Okay. Okay, next slide. Orunabo, go to. Okay, I am going to next. Right, I am because you are okay. Wait, I try to within 10 minutes. Okay, bear with us because lots of things not able to all hungry. So next time I will take class is responsible for discuss in details. So I think you are hungry, so time is already going to be ready. So I try to finish with not more 10 minutes with me. So in the biology, what I am for invention of biological materials, you have to submit the specification regarding filing the patent. According to, you have to submit the What microorganisms developed genetically modified microorganisms, in particular a depository center at first some Budapest Treaty. Next slide. Very interesting. You see 3 molecules, or rabiprazole, lansoprazole, omeprazole. You see, these are 3 molecules. The basic nucleus is same. Only change the side chain or side so, but efficacy is different. You see, slightly changes the molecule. New patents are coming. Omeprazole is different patent. Lansoprazole is different patent. Another is different patent. Rabiprazole, different companies file the patent on huge amounts of earning. Next slide. Already I told medical use patent is not allowable. Aspirin, aspirin is for headache and heart disease. If you find out another aspirin may be used for other disease also, it is maybe called new use of known substance. It is not patentable in India, maybe patentable in USA. Next slide. Method of treatment, already I told, method of treatment is patentable in EPO. EPO means European Patent Office, US Patent Office, but not in India. Next slide. Yes, gene sequence. You developed a new gene. Only gene is not patentable. If you use that gene, you insert that gene in the through RDA technology, you produce a vaccine or you produce a hormones, you produce form. Finally, on combination of all these things, new genes and method of genes produce the vaccine or antibiotics, all like that, all this together is a patentable service matter. Next slide. That I already told. Next slide. Transgenic, I told transgenic animals is patentable. In other countries, not in India. India microorganism is patentable. Next. Yes, if you see R&D process in pharmaceutical industry, lots of time is required for after the success of clinical trial phase 3. So it will take, if you find some molecules, some more than 10 to 12 days. After trial, it's a going for
So you are not audible.
not
Your voice is rapturing.
okay. Next slide, last slide. Go to last slide. All are last. Next to next, next. Okay. I'm not going to talk about PCTs. We'll take another time. Previous one. No, previous. Okay. If you see, no, next. Yes. If you see, all know what is generic medicine, what is brand medicine. If you see brand name medicine during its patent period, if patent period is over 20 years, provides good medical products at a low price, then they can use generic name, because after completion the patent period, it is no right for that particular patentee. So it is common for all. So it may be converted to generic medicine. Next slide. PCT, I am not going details. Next. Okay. Last time, what is the benefit of patent? Shall benefit the patent system, utilize resource R&D activities, capture global market, and profit through protected market. Next slide. Thank you. First, I am not happy due to some technical problem. So next time I try to explain that I hope I am able to give some idea.
Yes, sir.
Thank you all.
Thank you,
sir.
Dr. Dhananjay Saha, for a nice explanation on patents.
So
IPR.
any questions? Ask any question. Many things I not able to discuss. In future I will share with you.
Thank you, Dhananjay, for your nice explanation on patents.
Yes, Dhruvo, but the problem is that some technical is that for that sorry what has been happened
No problem.
really.
It happens sometimes.
But thank you, Professor Dhruvo Jyoti.
Welcome. You are most welcome. So this is
Thank you,
our
thank you.
book going to be published in Generous Publishing House by Australia.
It
Authors are
is yes. Previously, I told all of your students you are they are you undergraduate students. You so much giving inspiration to the student. They are doing good research publications, books. It's really example. I can boldly can say it is very rare you will not find out this type of achievements of the student. Those are doing books and publications in undergraduate. Even I can say when I was in undergraduate, I had no idea what is that time was also I had no idea. Started my PhD and slightly I learned how to do research. But you are giving input to the students how to do research. It's a really it will be great learning for the student future career. Okay, thank you to all.
Thank you. This is our book going to be published by 3 authors, myself, Dhananjay and Kushal from 3rd year. And the mega team project team members are Arunabho, Orgho, Sandeep, Kushal.
Sir,
Ah,
sorry to interrupt, but I want to that all team members of this book should open their camera and show their face.
okay.
All team members.
Now open.
Sure. Most welcome.
Sure.
চন্দ্রদা
হ্যাঁ
তুমি ভিডিওটা অন করো। চন্দ্রদা, সন্দীপ, প্রীতম, যারা যারা এখানে প্রেজেন্ট আছিস সবাই ভিডিওটা একটু অন কর।
Orgho, Pritam.
দেখা যাচ্ছে
So these are team members of our books. Chandra is present and other team members are Bornamoni Shaha, Anirbita Ghosh, Orgo Vishwas, Soumitra Ghosh, etc. Sir, you can continue now.
ওকে
Okay, Kushal, now announce for vote of thanks by Dr. Bera.
Yes, Kokon sir. Now I welcome Dr. Kokon Bera for vote of thanks. Sir, please continue. Kokon sir. Please continue.
এতটা ডিস্টার্ব হলো না মোবাইলে।
Dr. Khokon Bera, are you there? Dr. Khokon Bera? Dr. Khokon Bera, are you there?
Khokon sir, please unmute yourself.
Sir is coming,
Dr.
sir is
Khokon Bera,
coming.
are you
Yes,
there?
yes, sir, come.
Okay. Now I am going to speak vote of thanks. Myself, Dr. Khokun Bera, faculty of School of Pharmacy, Techno India University, feels immense pleasure to say something in this today's webinar in this Temple of Learning. TIU is hosting the scientific event for the second time in the history of Millennium. The theme of the seminar is protection seminar protection of world from culprit of ridiculous offensive nonsense air corona, which focuses on the pandemic situation of the corona in high altitude in the area of new drug discovery to combat on the various complicated physical ailments to be defeated by the pharma professionals working on various fields of academic as well as industrial level. Budding technocrats of pharmacy profession like diploma, degree, postgraduate, and doctoral degree holders are being motivated by current therapies subject to give outcome results on new drug discovery through various fields of pharmacy like pharmaceutical chemistry, pharmacology, pharmaceutics, and pharmacognosy, who are the four pillars of pharmaceutical science. Pharmacy is a subject which runs on its two legs, that is chemistry and biology, which is the key points to get success to give a fruit of drug to be compiled into medicine. The venture of this webinar is going to be fruitful outcome by the combined efforts given by our chairman and director, sir, Professor Dr. Bedouin Mohanty, and the teamwork of our respective faculties like Dr. Dhubajyoti Sen, Dr. Falguni Patro, Dr. Kokun Bera, and Dr. Bonkim Chandra Nundi as a group of conveners. And Orpita Vishwas, Susmita Bosak, Lavani Das, Suprati Mondol, Dr. Partharanjan Bhattacharjee, Shoktijit Das, Dr. Joyanta Das, Oswind Bhuiya, Dr. Dinako Sasmol, Dr. Tapon Kumar Bormon, and Dr. Gautam Bagji, Dr. Sajol Rai Chaudhary, Dr. Satvik Das, Sriporni Das, Tomal Das, Soumitra Pal, Gorhori Kamilla, and Sonu Srimanna as a group of co-conveners, as well as the energetic of enthusiastic technical assistance and social media influenced by Sondi Soykar, Kushal Nondi, Pritam Bokshi from the third year, and the fourth year is Arunabha Chandra Chandra, Sobhik Majumdar, and Soumadev Maiti. Anchoring blasters Kushal Nondi from the third year, Bornomani Saha, third year, and Shisha Majumdar from the fourth year have hired 2 days session. The exclusive of the teamwork led by Professor Dhubajyoti Sen of his dedicated venture in our motivation to get success with heartfully applies. Thank you all for my bottom of heart. Thank you. Thank you.
Thank you so much, sir.
Okay.
Now, I am going to welcome Dr. Dhubajyoti Sen for the final speech in this webinar. Dhruvo, please continue. Hello, sir. Are you there? Dhruvo, sir? Hello.
Okay. Okay, I am
sir.
here.
Sir, we cannot able to see you. Please open your video.
Oh my God. Hello, everyone.
Hello, sir.
I am here.
Yes, sir.
So, so this is today's second webinar of Techno India University, which has gathered tremendous responses from international and national level. I myself, Dr. Dhruvajyoti Sen, as a convener of this webinar, is very much happy to finish this venture nicely. And our all team members have supported me nicely in this motivation. This is the second webinar and the theme is protection of world from culprit of ridiculous offensive nonsense air, that is corona. C for culprit, O for R for ridiculous, O for offensive, N for nonsense, A for air, which has backed high tremendous impact response with fruitful outcome on the chariot of success. I appreciate the energetic and enthusiastic technical team and assistantship social media influencers like Sandeep Sarkar, Kushal Nandi, Pritam Bakshi from third year B Farm, and Arunav Chandra, Shovik Majumdar, Shaumadev Maiti from final year semester 7, anchoring blasters like Kushal Nandi from third year, Bornamani Shaha third year, Shisha Majumdar fourth year have fired this 2 days session, and 5-man army Dr. R. Badmanaban, Mr. Vedaduti Chakraborty, Dr. Bapaditya Chatterjee, Dr. Rutesh Vyas, and Dr. Dhananjay Saha made this webinar a fantabulous learning platform. Thank you all from my bottom of heart to make this fruitful outcome. Jai Hind. So this is our second day's webinar completed.
Thank you, sir. Sir, I think we cannot even to get reach college for 9 months for this COVID-19 pandemic. So we decided to play a video of our college at the
Okay.
last of this
No
session.
problem. You
So
can
can we play?
Yes, definitely.
Okay, Shandip, you can play.
This is our college visit. From one side to another side. Left, right,
So this is the video. It is made by Pritam Bokshi. So thank you, Pritam, for made this video.
Excellent. All the team members have supported me. Excellent. Thank you all.
Sir,
Thank
this is
you
the end
all.
of our session. Thank you so much.
Thank you all. Thank you.
Welcome.
Yeah.
So today's session ends here.